the protein structure and the levels from primary to quaternary. And then understand that the protein function depends on the folding patterns, and these folding patterns are because of the amino acid sequences and their molecular interactions. And ultimately, it's going to fold a protein, okay, based on those molecular interactions. Now, the guided question I want you to keep in mind today is that how does an amino acid sequence become a functional protein shape? So we have the amino acid sequence, okay? From the amino acid sequence, folding will begin because of the interactions between the R groups. The 3D structure would then start to take form, okay, and it falls into a very specific 3D shape. And then finally, that 3D shape might be the final form, or it can have a quaternary shape, but this 3D structure is the functional shape of the protein that has an active site or a binding site, for example. And then this leads to protein function. So in this case, this is an enzyme that speeds up chemical reactions, but it could also be a transport protein or a structural protein that provides support. The primary structure is the specific sequence of amino acids in a polypeptide. Okay, now amino acid 1, 2, 3, 4. Now changing the sequence can change the protein's shape. So the primary structure is the amino acid order. Here we can see the primary structure. Okay, that is the very first sequence. And as the interactions start happening, the protein becomes more and more complex. Now, why it matters, okay? So a change in an amino acid, if we have a single substitution, meaning one is changed for another, okay? So here we see one amino acid that is hydrophilic, meaning this amino acid loves interacting with water and forming hydrogen bonds. But it's changed for an amino acid that is now hydrophobic. Um, this amino acid does not want to interact with water. So it's going to be pushed inside of the 3D structure. Okay, so here we have the normal folding, but because we just have that one mutation of changing the sequence from hydrophilic to now a hydrophobic molecule, it alters the folding of the molecule. And if there is a change in shape, it changes the interaction and it changes the function. The secondary structure is to do with local folding, okay? And the local folding forms structures like alpha helices, okay? These twisty like structures and beta pleated sheets. Okay. Now, what is important here is that the secondary structure has to do with this N terminus and the C terminus and how they interact with one another. And they will form hydrogen bonds with each other. So the N terminus will form a hydrogen bond with the C terminus of another amino acid, okay? And this will form a hydrogen bond with the N terminus of another amino acid. Just to give a quick overview of the tertiary structure, okay? Now this is the overall 3D shape. And this is because of the R group interactions. And remember, each amino acid has a different R group. And there are four main interactions that you need to know about. You need to know about hydrophobic interactions, where the polar group is nonpolar, hydrogen bonds that happens through partial charges. So remember, this is like between the backbone molecules. The O is partially negative and the nitrogen is partially positive, so it's going to form a very weak hydrogen bond with each other. ionic interactions, which is when we have full charges, a positive or negative charge, and also disulfide bridges. Now, the quaternary structure. So we finished a tertiary structure, okay? But a lot of the times, some proteins are made of 2 or more polypeptide subunits, meaning it has a primary structure up until the tertiary structure, and then these tertiary structures would come together to form a functional protein. Now, the subunits are held together by the same types of interactions as the tertiary structure. Now, it's important to know that not every protein has this structure. Now, it's only quaternary if it's made of one or more, if it's made of one or more than, if it's made of more than one polypeptide subunit. Okay. And the quaternary structure is multi-polypeptides working together. To bring it all together, all levels contribute to function. Okay, the primary structure gives us the amino acid sequence. The secondary structure is the local folding, meaning how does these backbone molecules of amino acids interact with one another and form hydrogen bonds. Okay, it's the hydrogen bonds between the backbone atoms. The tertiary structure has to do with the R groups, and the quaternary structure is when 2 or more tertiary structures come together to form the final protein. Now, the shape matters, okay, because the protein function depends on the molecular interactions. So if the shape changes, the interaction changes, and then the function will change. Here is a type of biological scenario. So I have a protein that has a mutation and only one amino acid changes in that protein. So I have the original amino acid is a hydrophilic R group. Okay, so this one's hydrophilic. And now a mutation happens. After the mutation, I have another hydrophilic R group. So predict one effect on the protein's folding or function. So the protein may still function, it may still function the same because the new R group has similar chemical properties. Okay, so it would still interact with water and other chemical molecules in the same way. Now, a different type of scenario would be I start out with a hydrophilic R group, okay, but I change that out for a hydrophobic R group after the mutation. So predict one possible effect on the protein's folding or function. So this protein may fold differently because the new R group is hydrophobic. And this could affect the function by having different chemical interactions. And because it has different chemical interactions, it can change the shape. And if the shape changes, the interaction with other molecules would change, and that would change the overall function. If you enjoyed that video, please don't forget to like and subscribe, and can join us on the Skool.com community page where you'll find AP Bio blueprints. And if you take a look inside, you will see that Unit 1 is completely free. See you all there.